Hi and welcome to today's presentation on the history of microbiology as part of our introduction. And so the ancient people believed that diseases were the punishment sent from the gods for certain human indiscretions. However, man had already suspected that microbes were responsible for the decomposition of organic matter, also spoilage of our food, and some infections which were ongoing, and even the fermentation of alcohol. Now, we needed to develop. And so the science of microbiology started with the development of optical systems to view microbes. And so the most important device that transformed microbiology was the invention of the spectacle, which was by Hans and Zacharias Jensen tube of two lenses. Now, in 1665, a scientist by name Robert Hooke revealed that all living things were made of little boxes or cells. And this led to the cell theory. The theory states that all living things are composed of cells and come from pre-existing cells. And this also supports the germ theory, the idea that diseases arise from transferring and replicating microbes and pathogens. And so if someone has, for example, tuberculosis and is coughing all over, and someone who doesn't have the disease catches these microbes from the air, the microbes enters the system and begins to multiply or replicate and cause disease in such a person. But it was Anton van Leeuwenhoek who described the live microorganism and called it animalcules. So these were the initial stages of the development of microbiology. Now, during this time, people also believed that life actually, could arise randomly. You understand? And also emerge out of non-living matter if certain conditions or ingredients were met. For example, maggots were thought to generate themselves out of rotten meat, and also worms were thought to emerge from the combination of soil and water. It was interesting, isn't it? But now we know that maggots emerge from eggs which are laid by the parent flies. And interestingly, if you've observed closely, you find that lots of worms appear after rainfall. It's now common sense that the worms stay underground because their skin needs to be moist so that they can breathe. Now, when it rains, the ground is covered in water, so they are free to move about. And so these were some of the thinking, and this now is what we've come to know. During this time also, people used to believe, that worms, rather than hatching from the eggs of other worms, were created when dirt and water made mud. So this was the idea of a popular, theory, which we call the spontaneous generation. And so in the 17th century, two main theories were entertained. The first theory being spontaneous generation, and the second being biogenesis. Now, when we say spontaneous generation, is an absolute theory that states that living organisms can originate from inanimate objects. Other common examples of spontaneous generation were that dust created flies, maggots arose from rotten meat as we've seen before, and that bread or wheat, when you leave it in the dark corner, can produce mice. These were the thinking, and these were the thoughts of the proponents of spontaneous generation. Now, during this time, many experiments were conducted to both prove and disprove the theory. But before that, we will look at the other theory, which was biogenesis. And as we know, biogenesis refers to life from life. But bio, if you break the term, bio means life, and genesis means the beginning. And so literally, biogenesis should mean the beginning of life. And so the proponents of biogenesis also stated that life can only emerge from living organisms. And so now we've gotten to know that they were describing reproduction, which may either be through sexual or asexual reproduction. Now, like I said, lots of experiments were conducted to prove and also disprove, these theories. So we are going to look at some of them and what transpired over there. So the first scientist who set out to disprove spontaneous generation was Francisco Redi. And what he did was he put fresh meat in an open container, which was a decaying meat anyway. So the decaying meat attracted flies onto itself, as you can see over here, and the meat was soon covered with maggots. And the maggots also hatched into new flies, as you can see in the open jar over here. Secondly, he covered the whole meat tightly so that flies could not get into the container. And what was seen was no maggots were produced, as you can see, so it was tightly closed. Then finally, he covered the jar with layers of porous gauze. And since the flies could not enter, the flies were rather attracted to the smell of the rotten meat. And so they clustered on the gauze which were covered, which were used to cover the jar, and was soon, filled with maggots on top, as you can see over here. But the meat itself was free from maggots. And so what he said was flies are necessary to produce flies. And so living things are necessary to produce living things. They do not generate by themselves. So the first scientist to disprove spontaneous generation was Francisco Redi. And what he did was he put fresh meat, which was almost decaying, into an open container, as you can see over here. The rotten meat attracted flies onto it, and the meat was soon covered in maggots, as you can see in the lower container. These maggots later hatched into flies again. He repeated the experiment, but this time he covered the jar tightly such that no fly could perch on it. And so eventually there were no maggots generated on the meat. Then finally, he covered the jar with a porous gauze. But since air could enter, the flies were attracted to the smell of the rotten meat. And so they clustered on the gauze and maggots were produced. But the meat itself inside the container was free from maggots. And so Redi concluded by saying flies are necessary to produce flies. They do not arise spontaneously from the rotten meat. So this is what Francisco Redi did to disprove the spontaneous generation theory. However, another scientist by name John Needham set out to also prove that actually spontaneous generation was the order. Now, everyone knew that boiling killed microorganisms. However, Needham wanted to find out if microorganisms appeared spontaneously after boiling. And so what he did was he tried to disprove Francisco Redi's conclusion. And so he boiled a chicken broth. Then he put it into a flask, sealed it, and waited for some time. Now, after a while, he saw that the broth was filled with microorganisms. And so he claimed victory for spontaneous generation because he said there appeared microorganisms from nowhere. So they appeared by themselves. Now, Needham's experiment was scrutinized by his scientific peers. They argued that he may not have boiled the broth long enough, or that the broth was introduced to new microbes even before he sealed the container. And so another scientist by name Spalanzani also set out to attempt to refine John Needham's experiment in order to disprove spontaneous generation. So he repeated almost everything that John Needham did, but there were just two differences. What he did was he boiled the broth for long in order to kill all the microorganisms, and immediately he covered the bottle, or he covered the container. So as you can see, he boiled the first one, which was here, left it open, and he realized that after some time it was filled with microorganisms. The second time, what he did was he boiled the broth, then immediately covered it, and it stayed there for so many days, but still no growth of microorganisms was seen. And so he also claimed that you needed living organisms to produce living organisms. However, the scientific community once again, dismissed Spalanzani's experiment because they argued that he excluded air, which was a requirement for spontaneous generation. And so finally, Louis Pasteur set out to disprove spontaneous generation once and for all. What did Louis Pasteur do? He boiled, first of all, he boiled broth in a swan neck shaped flask, and he tried to boil to the extent that all living microorganisms died. After some time, he left the broth to sit for a while, and he stayed for days, but there were no bacterial growth. That wasn't all. He did the second test. He repeated the test, and this time he decided to break off the neck of the S-shaped flask. So he broke off the neck and allowed it to sit for some time. This time around, air entered the flask, and so after a while there were present of bacteria in the broth. Finally, Louis Pasteur decided to rather tilt the glass because he realized that microbes were collected at the neck of the flask, but they were unable to enter the broth. So he tilted the flask and allowed it to sit for a while, and bacterial growth were seen. And so what Louis Pasteur did was to prove that you needed microorganisms to enter in order to cause other microorganisms. So Louis Pasteur set out to finally disprove spontaneous generation. And what he did was to bring the whole debate between spontaneous generation and biogenesis to an end. Now, Louis Pasteur also proved why beer and wine usually sour. And so he explained that yeast will convert sugar into alcohol without air, and he called that process as fermentation. But he rather said, in the presence of air, the alcohol is converted to acetic acid by bacteria. And so he demonstrated that these spoilage, bacteria could be killed by heating them for quite a short Wow. And this was going to evaporate the alcohol, in the wine. And so he proposed, or the term pasteurization was named after him, as the application of a gentle heat for a short time, which killed most bacteria in any substance that you applied. So, Louis Pasteur showed that disease can also arise when microbes entered tissues and grew. And this, as we saw earlier, supported the germ theory. And so we also had the pasteurization of milk. So we use the same procedure to get milk to be safe for drinking without any microbes. And so with the pasteurization of milk, the milk is first heated to about 70 degrees Celsius for about 15 to 30 seconds. Then it is immediately, chilled and stored. And this process actually deactivates microbes and also reduces enzymatic activities in the milk. Now, after Louis Pasteur, we had John Tyndall, who also supported Louis Pasteur's theory and said heating the broth decreased the spoilage. And so with this process, we got to know about the process of sterilization. Now, John Tyndall discovered that there were two types of bacteria. He said some of the bacteria were sensitive to heat, or they were heat labile, and others were resistant to heat. Then he said the ones which are sensitive to heat are easily destroyed by boiling, but the ones which are resistant to heat are called the endospore forming bacteria. Now, he demonstrated that alternate processes of heating and cooling, if repeated 5 times, can kill all endospores. And so the term tenderization is the same as sterilization, and is the process of rendering all surfaces and also instruments or materials safe from microorganisms, including the bacterial endospore. Now, Robert Koch was another scientist who came, a German physician, who developed the criteria known as the Koch's postulate. Now, what did he do? He said, or he was also among the first to isolate bacteria in pure culture. And he said that for us to establish the cause of any disease, we must, or we should be able to identify a specific microorganism causing that disease. And so, as part of his works, he was able to actually isolate bacteria, including the Mycobacterium tuberculosis. Hence the name Koch's disease being referred to as tuberculosis. Now, let's look at what he postulated. He said, so we have 4 main things under the Koch's postulate. The first thing is, for we to say someone has a particular disease, the microorganism which is causing the disease must be found in abundance in all organisms who are suffering from the disease. But this microorganism should not be found in any healthy organism. And so if we have A to be having tuberculosis, if A is having TB, then we expect that A should have the Mycobacterium tuberculosis in his blood. But B, who is not having or tuberculosis, should not have this Mycobacterium tuberculosis in the blood. Number 2, he said the microorganism which is causing the tuberculosis must be isolated from the diseased organism and grown in pure culture. So we should be able to isolate the Mycobacterium and grow it in the laboratory. Now, the third postulate is that the cultured organism or the cultured microorganism should cause disease when introduced into a healthy organism. And so it was A who was having the tuberculosis. When we introduce this microorganism that we took That is the Mycobacterium tuberculosis. If we introduce it into B, we expect to find Mycobacterium tuberculosis in B as well. And so B should fall sick with this microorganism. And finally, what he said was the microorganism must be re-isolated from the inoculated diseased experimental host and identified as being identical to the original specific causative agent. This is very simple. So he said that once we've introduced the microorganism into B, for B to also get tuberculosis, when we isolate or when we take samples of B, we should be able to identify that, or we should be able to prove that the microorganism found in B is the same microorganism which was found in A causing the disease. Now, the Koch's postulate rather does not hold for certain infections. For example, it doesn't hold for HIV, oncoviruses, co-infection with Staphylococcus aureus, and also some opportunistic fungal infections, as well as Candida albicans. All of these do not follow the Koch's postulates. And so, to describe this with pictures, we have a diseased animal over here, and this is a healthy animal. There's nothing wrong with him or with it. So we took the cells, or we took samples of this diseased animal. We observed it under a microscope, and this was what we found. Now, this red blood cells, when we grow it in the laboratory, it should show something like this, but this healthy animal shouldn't have any pathogens in his blood. Now, we are going to introduce these microorganisms that we isolated from the diseased microorganism into the healthy microorganism. And when we do that, it should cause disease in the healthy animal. Now, afterwards, when we take samples of this animal, we should be able to identify, or we should be able to see that it is the same organism that we transferred from the diseased animal that is causing the disease in this new animal. So this is all about the Koch's postulate. Now, let's also look at what culture medium is. And so we use the culture medium to isolate microorganisms. It helps us to identify one type of organism. It was very, very important during the Koch's period. And initially, the culture medium that we were using was the gelatin. It was the first, that was being used. But recently, it has lost its usefulness because it solidifies certain agents when it melts, and also it melts at temperatures more than 28 degrees Celsius. And also some bacteria were hydrolyzed with its enzyme, and so it's recently, or of late, not being used as much. However, we had other scientists who developed new culture media, and we got to have the agar. And this is advantageous because it does not digest or hydrolyze the bacteria, and it can only melt at temperatures more than 100 degrees Celsius. And we use it much in today's period. But we also had Richard Petrie, who was also assistant of Koch's, and he developed the Petri dish, which we use now. And so this is a brief summary of the history of microbiology.